that it also can be in symbiosis with some fungi and other algae as well, together with corals. Okay, if you look at coral, it's a symbiosis between that particular polyp coral and also an algae called zooxanthelle. Okay, so the algae provide oxygen to the zooxanthelle, the algae, which is actually a zooxanthelle or diatom. It provides oxygen to the polyp, the coral, and the coral provided some refuge, okay, safe environment. So it's a symbiosis, okay. But most of the microalgae essentially, living freely without any kind of attachment, or rather in being parasitic to other species. So this is the role of microalgae on what do you call this, in aquaculture. All right, now take a look at this picture. I hope you can see it. It's not very clear, but I think you can nonetheless see it. Like, I tried to reproduce this before, but it's a bit difficult. Okay. So let me just show you what we have here. Okay. Now, if this is a face-to-face meeting, I will draw it on the board. You see. Okay. Face-to-face meeting, I'll do it on the board. Anyway, I will just go through quickly so that you understand what are the roles of microalgae in relation to aquaculture. Very important, in relation to aquaculture. There are many other roles outside there, but this is more in relation to aquaculture. Give me a second. All right. If you look at this round circular picture here, you see the word microalgae. Can you see this word here, microalgae? Boleh nampak tak tulisan ni? Can you?
Yes.
Boleh je lah.
Boleh? Okay. So you see here microalgae and bacteria down here. There are all sorts of microalgae and bacteria. Now these two are live food organisms. As I mentioned earlier, you can put in or add bacteria as also a live food organism because it's alive, right? But in our case, we are more interested in the microalgae for the time being. Okay. All right. So I got, just give me a minute or so. I'm trying to get some things organized here quickly. Yeah. Sekejap. Okay, so you have this microalgae, and then you look at this picture here. This is the early, okay, and late mollusk larvae. Mollusk, M-O-L-L-U-S-C, mollusk. What are mollusks? Mollusks are essentially also an invertebrate, okay, kerang-kerangan, mollusk, okay. Early and late mollusk larvae, and then here you have adult bivalve mollusk. Now, when you look at microalgae, you look at this arrow here. Now, the microalgae is food for the mollusk larvae, all right? And it's also food for the adult mollusk. Let me say this again. The microalgae is given as food or consumed as food by the mollusk larvae and also by the adult mollusk. Okay, molluska yang dewasa dan benih molluska, bahasa Malaysia kadang-kadang digunakan istilah rega molluska. Dua-dua memerlukan microalgae. What does this mean? It means that if you have a hatchery, that produces your mollusk, example, hatchery to grow oyster, okay? Hatchery to grow oyster and grow out of oyster, and nursery of oyster, then you need to have your microalgae being produced all the time. Now remember, I'm not sure that you know this or not, or you remember this, or have anybody has taught you about this or not. In aquaculture, you have basically 3 production areas. Dalam aquaculture ada 3 merupakan 3 sudut pembesaran, ya. Okay, 3 areas of production. One is the hatchery, satu di hatchery, satu lagi di grow out, pembesaran. So di antara hatchery dengan grow out, apa ada kat tengah-tengah tu? What is it? In between hatchery and grow out. Anyone knows? Anyone knows? Between hatchery and grow out, there is one other component there which is also very important. What is it? Anyone knows? Bahasa Malaysia dia panggil asuhan. In English?
Nursery.
Nursery, yes. So, you will have hatchery, nursery, grow out. Grow out tu pembesaran. So, this is the adult for grow out. This is the hatchery to produce your early mollusk larvae, and between here, sometimes you have what we call as a nursery. Okay, so for your mollusks from the hatchery production to the nursery to the grow out, at all stages, they require the supply of live microalgae untuk mollusk. Alright, so remember this. Kalau you dalam projek menghasilkan molluska, oyster for instance, or mussels, kupang, okay, if you are in the business of growing those, you must grow your microalgae at all stages, from the hatchery to the nursery to the grow out. That is for mollusk. So what about for shrimp? Here, shrimp. Okay, for shrimp, the food here, your microalgae, all right, is given to your copepods, rotifer, or your brine shrimp. What is brine shrimp? Brine shrimp is Artemia, okay? So you give your microalgae as food to your zooplankton, and then your zooplankton will then be fed to your shrimp. Okay, all right. So some of the nursery also, some of the shrimp larvae also, not some, rather all shrimp larvae also will require microalgae. So it's quite unique, this shrimp here. At the larval stage of the shrimp, it requires microalgae at some point. And then as it undergo metamorphosis, as it becomes bigger and bigger, it will require zooplankton such as brine shrimp. So depending on the stages of growth for the shrimp, the earlier stage is normally the microalgae, and the later stage of larval development, they require zooplankton such as brine shrimp or Artemia. Jadi udang ni pada peringkat awal, makanan pertama adalah microalgae, disusuli oleh zooplankton seperti Artemia, dan kemudian boleh dibagi makanan rumusan or processed feed sampai besar. So different ya, berbeza ya, different between shrimp here and mollusk. Mollusk, all stages you require microalgae, but for the shrimp, the earliest stages, very early, you require microalgae, but later on, you supply the zooplankton and switch later on to processed feed, makanan rumusan. For mollusks, you don't give processed feed. All right. So what about fish? Fish also the same. You give microalgae, all right, to the, what do you call this? The larvae of your live food and then give to your fish as well. This is the early larvae of crustacean and fish. It requires some microalgae too. Some requires microalgae. Some can feed on the zooplankton. Okay, so fish and shrimp are somewhat similar. Okay, but in most cases, the marine fish, they can take directly the zooplankton first. No need to give them microalgae. Okay, but the zooplankton will require microalgae to feed on and grow. So these are some of the differences in terms of how the microalgae plays a role, especially in the production of larval fish, larval shrimp, and larval mollusk. Boleh ya? Alright. Now, in a fish or a shrimp hatchery, you'll find that there are a few components. Beberapa komponen dalam hatchery udang dan hatchery ikan. Alright. Now, the first thing here you see, so this is an example, let's say, of a fish hatchery or shrimp hatchery. Okay. Now, the first circle here, all right, is called maturation. Okay. Maturation. Now, what is maturation? Anyone knows what is a maturation? You can imagine this as tanks. Ini tangki ya. Tangki warna biru kecil, tangki hijau dan kecil, tangki hijau besar. You can imagine this as different tanks, all right? Okay, so you look at the arrow going this way, this way, this way, this way, this way, this way. All right, so what is maturation? Any idea? What is maturation?
Grow up tank.
Not really. Quite, okay, you're close. There are some points there, okay, but not really. Maturation. Kematangan. Atau you can call it in bahasa Malaysia, tangki pematangan. Apa yang nak dimatangkan? Tangki pematangan. Maturation. What is it that you want to mature? Apa yang nak dimatangkan?
Maybe they able to reproductive.
Yeah, okay. So when you say maturation, it means you want to mature the brood stock. Kita nak mematangkan induk. Okay. So remember, in a hatchery, to produce the larval fish or larval shrimp, you need the brood stock, induk. Yeah, you need induk. Kita perlukan induk ikan untuk udang untuk menghasilkan rega atau benih udang atau ikan. So maturation means, atau pematangan maksudnya, it means that you want to mature the broodstock so that they are ready to spawn. Okay, you want to keep the broodstock in the hatchery, in the maturation tanks or maturation room, so that they are ready to spawn. What is spawning? What is spawning in bahasa Malaysia? Apa maksud spawning? What is spawning? Any idea? Anyone?
Persenyawaan.
Persenyawaan is mating.
Bertelur.
Bertelur kan?
Yeah, peneluran, spawning. Senyawa is fertilization sebenarnya. Persenyawaan is fertilization. Spawning is penghasilan telur. Okay. spawning, your egg production. Simply said, egg production. So once the broodstock are mature enough, they will mate and the eggs will be fertilized. Disenyawa, okay? Once the eggs are fertilized, at some point it will hatch, right? Now that egg production and hatching is called spawning. All right, so as after the eggs have been spawned, then you will take the larval fish, or sometimes you take the egg also that has been spawned and put it inside the larval rearing tank or tanks to grow the larvae. Ikan-ikan yang menghasilkan telur, telur ini diambil dan diasuh di dalam nursery. Remember I said earlier, hatchery, hatching. The word hatchery comes from the term hatching, hatching. This is why I call it hatchery. All right. And then when the eggs hatch, you transfer it to the larval rearing tank or larviculture tank. Tangki untuk asuhan larva. Ini. This one. Now in the larval rearing tank, now I prefer to use the word food. Live, instead of live feed, we use live food. Food. That would be a better term lah, okay? Feed is normally formulated feeds, but food is more, can be consumed live. So you add the live food to this larval nursery tank, and then you will grow it or nurse it until a certain stage before you take it out from the hatchery or from nursery and take it to the grow out pond, grow out tanks, and so on. Okay, so remember the key areas here: maturation of broodstock, mating happens, eggs fertilize, they spawn, okay, production of eggs, hatch, you transfer the eggs to the larval or nursery tank. And then once the larvae have absorbed their yolk sac, now remember yolk sac, right? Precocial larvae, altricial larvae, we spoke about this earlier. Once they have absorbed their yolk sac, you have to give live food here. If not, they will die. As simple as that. So the timing is very important, right? So you grow the larvae, you nurse it until a certain stage. When they are ready to be transferred out to the grow out pond. Okay, so this is basically the key component in a shrimp or fish hatchery, right? You do have other things like this, quarantine, acclimation, which I have not said before, but essentially if you have your broodstock coming from other places, let's say you want to start your maturation, or your broodstock arrived from somewhere else, you just brought in your broodstock, you need to quarantine the broodstock first just to make sure it is free from disease and your broodstock is not stressed. If they are stressed, they cannot mate and cannot mature, of course. They can't mature, definitely. Yeah? And then you need to acclimate to your condition in the hatchery. So this too happens, especially when you bring in new broodstock. If the broodstock has been in your hatchery for quite a while, you have been keeping the broodstock for a long period and they're used to your water conditions and so on, there you can mature it quite easily. Now, how do you mature your fish? How do you mature your shrimp? Well, sometimes by putting male and female together, they get excited and then they mate. Sometimes you need to use, you need to inject them with some hormones, right? Right. You need to use artificial hormonal reproduction methods, all right? Sometimes you need to change the temperature, change the lighting condition, and so on. So all these are ways and means to mature your broodstock, to get the eggs mature, and so they can mate and spawn. All right. So it depends on the species that you are talking about. So when you talk about microalgae, there are major classes and genera of cultured algal species. Today, there are more than 40 different species of microalgae. They've been isolated in many parts of the world and to be used as your food or live food organisms. All right. So essentially, there are 8 major classes and 32 genera of cultured microalgae. We will learn more about this in the lab. Now, in the lab, what does that mean in the lab? It means that once we can meet up face to face, then you can see all the major classes of microalgae. All right. Well, you can see pictures of them, no problem. But then you want to see them, the color of the microalgae, the density, how it looks like, and so on. It is best that we look it under the microscope, okay, and you can see all of them very clearly. Right, so these are the most frequently used species in commercial mariculture. Mariculture means what? Aquaculture in marine environment. So some people use the term mariculture. Mari means marine, seawater. Okay, so mariculture operations for for your shrimp and your fish. Okay, marine shrimp, marine fish. So diatoms, you can use species such as Chelitonema, Thalassosira, all these things. Flagellates, remember they have the flagellates, okay? Flagella, just like that plankton picture you saw in the beginning, right? And then these are some species of flagellates such as Isochrysis, Tetraselmis, and of course your green algae like Chlorella. Now we have stocks of this. We here in UPM do keep stocks of diatoms and flagellates, not here in Serdang, but we do have some samples of this in our station in Port Dickson. We have our station Perikanan in Port Dickson, or marine fish station in a marine research station in Port Dickson, where we keep some of this microalgae. Now, my question to all of you, have you been to our Center Marine Research Center, Marine in Port Dickson? Have you been there?
Not yet.
Not yet, huh?
Not yet.
Not yet. Okay, you have not been to our, we call it Center of Marine Science or Research, something like that, okay? Okay, sometimes they change names so often I get confused. But anyway, we do have our Marine Fish Research Center in Port Dickson. Okay, let's see whether maybe we can go there one of these days for you to take a look at our station there, okay? Now, this is a collection of diatoms. I just want to show you so that you can see the various kinds of diatoms here. If you look under the microscope, you can see all of this. And they come in many forms. They come in spindle shape like this, round shape, and so on, okay? And they're very unique in the shape is quite distinct, all right? And you do have some flagellates like this, remember? Flagellata, flagellates here. Okay, and you have Chlorophyceae, example, some of the chlorophytes or green algae, some unicellular, single cell, but flagellated like this, Chlamydomonas and Chlorogonium, all those, okay? But the Chlorella is not flagellated. You know, Chlorella is a Chlorophyceae or Chlorella, but it doesn't have any flagella, just round shape Chlorella, okay? Spirulina is spiral shape. That's why I call it spirulina, spiral shape, okay? Now, these are other kind of microalgae. You have this Chaetoceros, this is Tetraselmis, this is your Dunaliella, and also Chaetoceros. This is Chaetoceros is a diatom, all right? Now, you might get confused at this point when you see all these photograph of microalgae. In the lab later on, I will tell you what are the more important ones. Okay? Not to worry at this time, because once you go to the lab, I'll show you the different kind of microalgae, the important ones. Then it's easier to understand. Okay. Now, these are some of the more important microalgae group, okay, that we need to be aware of. Again, you don't need to memorize this. No need to memorize. Okay, just to tell you that there are different classes. Bacillariophyceae, this is the diatoms. Haptophyceae, you have the isochrysis, chrysophytes. Prasinophytes, where you have tetracell mist just now with 4 flagella. You have Cryptophyceae like Chlamydomonas, Cryptomonas, Rhodomonas, all this. Chlorophyceae, okay, like your Dunalela just now, and Cyanophyceae like Spirulina. So these are basically the different classes of microalgae. And then you have the example of how they are used in aquaculture. This, okay? All right, now, as I said, you don't need to memorize this, but when we do our lab, later on, when we do our lab, I will give you examples of like Bacillariophyceae. Maybe we have Skeletonema or Chitoceras. So give example of this. And Haptophyceae example. Cryptophyceae example. So from the examples, you need to understand how they are used in hatcheries and so on. Okay. Now those things in the lab, where we give you that, you need to understand and have some memorize some of them as well, but not this particular table. All right. So just to let you know. So unfortunately, we can't. So the first week of November. I think when we all meet later on the 8th, I think after the 8th, then we will see the different kinds of microalgae as well. I will have to also compress some of the classes. So we'll be missing 2 or 3 labs because of this MCO, or because of the restriction, COVID restriction. But once we start meeting, some of the labs I will compress, meaning that I will join 2 or 3 labs together at one go. Okay, or what we have missed, we cover during the 3rd or 4th week of lab. Okay. All right. When you look at microalgae production, there are very important parameters to look at. Okay, one is the nutrient quantity, how much nutrient that you need to give to your microalgae. The other one is nutrient quality. All right. Then light, very important, right? Because microalgae, it has to go photosynthesis, you need light, all right? pH is also key, because if the pH is too high or too low, they will die. The pH should be around neutral or slightly more than neutral. You need aeration. If you don't have enough aeration, they will die also because of what we call as self-shading. Self-shading, they will shade one another, especially when the light, when they are very high in abundance. Salinity, kemasinan, very important also, depends on the species. Some species require fresh water, some marine, but some are what we call as cosmopolitan, meaning they will grow in both marine and fresh water. Okay, like chlorella. Chlorella you can find in fresh water, and you can also find them in marine. Okay, brackish water as well. But another microalgae such as Nanochloropsis, you find them in marine, but not in fresh water. Spirulina, fresh water. So it depends on the species. Yeah? Temperature also another important factor to look at. All right. Now I will go through the factors first, and then later on I'll show you some pictures now. Okay. This part is a bit boring, I understand. But just understand that when we grow microalgae, we need to take care of these key parameters. The quantity of the nutrient, the quality of the nutrient, so they need nutrients to grow, the light, pH, you need to have some mixing, aeration, salinity should be right, and also the temperature as well. All right. Now, when we talk about microalgae, the first key the most important part is the nutrient, where you know that microalgae, for them to grow, you need nitrogen and phosphorus for cellular processes. All right. So all of them need nitrogen and phosphorus. That is why when you grow fish in your tank, or rather outside in your fish pond, let's say you have a fish pond, and you put your fish in there, you'll find that in the beginning, you will find that the water may be clear, right? The water in your fish pond is quite clear. You can see the fish. But as you feed the fish, okay, and then as the fish defecate, okay, or poop in the ponds, and then you'll find that the water slowly become greener and greener. Now, the fish poop contains a lot of nutrients such as nitrogen and phosphorus. And the fish feed also that you give to the fish contains nitrogen and phosphorus. So all this will encourage the growth of phytoplankton inside the fish pond. So that is why when you grow fish in a pond, the water will slowly become greener and greener and greener, and you give too much feed, it becomes too green and become eutrophic. All right. So all this because of the nutrient. So similarly, when you grow your microalgae in hatcheries, you need to make sure there is enough nutrient, especially nitrogen and phosphorus. In addition to this, you do need a particular element called silica, especially for diatoms such as Chaetosaurus and Skeletonema. Why silica? Because silica is used for the growth of diatoms, all right? And then they use this element, this silica, okay, production of their shell. And this diatom also is used or is consumed by shrimp, especially shrimp, udang. Now, udang needs diatoms because the shrimp shell, one of the components of shrimp shell is silica. And they get silica from eating or consuming the diatoms. So remember, when you grow shrimp, you need some silica as well. When you grow fish, you do not need silica. But shrimp, yes, you must have that. If not, they cannot grow, they cannot undergo molting. This is a bit interesting, especially for when you prepare the nutrient, diet, or formulation to feed your microalgae. All right, so other than that too, nitrogen, phosphorus, silica, you do need some micronutrients such as B complex, vitamins, all this. All right. Now, when you grow your microalgae, you do need to be aware of that some of the media that you prepare to culture your microalgae, all right, can be expensive. Okay, media yang kita gunakan untuk menghasilkan microalgae kadang-kadang boleh jadi sangat mahal, ya, sebab dia perlukan bahan kimia yang pure ataupun yang tulen. Alright, so when you grow your microalgae in lab, it is good to have what we call as lab grade or laboratory grade fertilizer. Apabila kita menghasilkan microalgae di dalam makmal, kita perlu menggunakan bahan baja yang dipanggil grade makmal, laboratory grade. Kenapa? Kualiti dia tinggi, tidak ada pencemaran atau kotoran. Alright. So if you are growing your microalgae in the lab, it is important that you use laboratory grade fertilizer or nutrient which is not contaminated, alright, tak ada kontaminasi dengan kotoran ataupun bakteria dan sebagainya. Right? So that is in the lab. So when it goes out of the lab, you can add in or put in what we call as agriculture grade fertilizer. When you talk about agriculture grade, you can buy it. It is quite cheap, okay, but it is okay to buy the agriculture grade for large scale production, because you don't need it to be very clean or very or free from contamination. But in the lab, you must make sure it is free from contamination. It is why the media that we use, it can be quite expensive also. Okay, alright. Kalau kita nak naikkan microalgae dalam kolam udang, kolam ikan, okay, if you want to increase the population of microalgae in fish pond, in shrimp pond, we just add in fertilizer that we buy from the store. Simple fertilizer. Okay, you can buy urea, which has a lot of nitrogen. Okay, you can buy maybe CIRP, Christmas Island Rock Phosphate, to give it the phosphorus. You can use all this. Okay, agricultural grade fertilizer to grow your fish in the pond. Okay, which is exposed to the sun and everything else. But you cannot use all those kind of fertilizer in the lab. In the lab, you have to buy graded or pure fertilizer, okay, elements. So this is an example of the fertilizer that we can use for mass culture of marine algae. Mass culture, what is mass culture? Mass culture means you culture in a big quantity, right? When you grow microalgae, in some cases you need to mass culture them, grow in big quantity, right? Which is outside of your lab, where you grow the microalgae stock culture or the beginning culture. So you have the small, you don't call it small culture, you call it the inoculum, or you start with the stock culture, the beginning culture. After that, you will scale up and then do mass culture. For mass culture, as I said earlier, you don't need to use lab grade materials or lab grade chemicals. You can use this, such as ammonium sulfate, which is ammonia contains nitrogen. You can use urea, which contains nitrogen also. You can use calcium superphosphate, which contains calcium and phosphorus. Clewat 32 is a brand that contains nitrogen and phosphorus too. And NP fertilizer 1620, NPK 162020, all this. This is essentially fertilizer for your plants. But you can use this as well for fertilization to increase your microalgae concentration. Okay, so no issue with this. You can even use any of the fertilizer that contains nitrogen and phosphorus to encourage growth of your microalgae, because microalgae also requires nitrogen, phosphorus, potassium, all this. All right. What about light? Well, light, you need to have a minimum of 18 hours per day. That will be good. All right. 24 hours per day, 18 hours should be lighted. The rest 6 hours. It's okay to get it to get it darkened. But sometimes you want to use just 12 and 12 hours light, 12 hours dark. All right. So if you can manage with 12, it's okay also. But normally we should go for 18 hours of light per day. But the light should not be too strong. If it's too strong, then you will actually cause some problems to your microalgae. All right. You can actually kill the cells of the microalgae if your light is too strong. Okay, kalau cahaya terang sangat, kita boleh membunuh microalgae cell. pH, well, as I said earlier, pH should be around 7. You want to range it between 7 to 9. But 9 is quite high, isn't it? They say, tingginya cik lah. No, it's all right. 9 is fine. Because the pH of seawater is around 8.3 to 8.5. You go up a bit high to 9 is still okay, but not too much higher than 9. Okay. In some cases, if you want to correct the pH, if it's too high, you want to add carbon dioxide to reduce the pH. All right. So why add carbon dioxide? Well, carbon dioxide is an acid in water. Kalau air dalam ternakan mikroalgae itu pH dia tinggi, kita letak karbon dioksida. Karbon dioksida itu adalah asid dalam air, jadi mengurangkan pH. Alright, so this is one way where you can encourage the growth of microalgae, because number one, the CO2 will drop the pH to a more desirable level, not too high. At the same time, CO2 is also used by the microalgae for photosynthesis. All right. Aeration and mixing, very important also. You want to make sure you do not leave it standing. These are the reasons for mixing: prevent sedimentation, to ensure the cells and population are equally exposed to light, to avoid thermal stratification. Thermal stratification is, in bahasa Malaysia, we call it perlintapan terma. Perlintapan terma, especially in outdoor culture where the pond is very deep. Okay. And then you want to make sure so that when you use mixing, you will improve gas exchange between CO2 and the culture medium and the air. All right. But to me, when you do mixing, the most important part is 1 and 2. To prevent sedimentation of the algae, number 1. Number 2 is to ensure all cells of the population are equally exposed to the light and nutrient. This is especially in the lab. In the lab, when you use, all right, when you use culture in the lab, number 1 and number 2 is very important. If not, the microalgae will die. You need to make sure that it's moving all the time. This is called aeration and mixing. Temperature, well, it's good to have 18 to 24, but not too low, below 16, not too high after 30. Of course, when you have a lower temperature, it will slow down the growth. A higher temperature would, of course, increase the growth with this. We will learn more about this in the lab later on when we use a different kind of microalgae species to look at. Then I will let you know what are the salinity that is best for them with the fresh water or marine or brackish water. All right. So essentially, whatever I've said just now, we can cover with this table: your temperature range, your salinity range, your light intensity, your photo period, and then your pH. All right. Now, I want to stop there for a while. Let's have a breather for a short while, and then when we come back, I will show you something in relation to the growth of algae as you start growing them in the lab. All right? Okay, so why don't we take a quick break? Boleh, guys? Hello?
Boleh.
Boleh.
Good. Take a think this just now. Sekejap. There you go. Okay. So this is essentially the growth phase of microalgae. All right. So when you grow microalgae in the lab, you will see this sort of growth phase. Okay. It may not be exactly like this, but it will be somewhat like this. You know, maybe it will start slower or later, and so on and so forth. All right. So you look at this, the growth phase. There are 5 stages, all right? Now, it is not exactly, as I said, very smooth like this, but it can be somewhat like this, you know, and then go up, come down, up and down, and in the end it will die, all right? So is someone joining the class? Zul Haikal, there you go. Okay, Haikal, welcome to the class.
Yes, okay.
All right. Right. So as I was saying, you have these growth phases, right? And hold on. It starts with what we call as a lag phase. Okay, lag phase or induction phase. Okay, jadi from there you go to exponential phase. Number 3 is the declining growth. Number 4, stationary phase. Number 5 is death phase. So if you look at how they derive this, this essentially this trend, you look at this on the left is the log of cell numbers, the density, cell density essentially, and this is the age of culture. All right. When you compare the log of cell density, which is essentially the growth rate, and you compare the age of culture, you'll find that in the beginning, your microalgae will grow very slowly. Look at this, phase 1. Let's say you put in your microalgae in a container. Okay. You put your microalgae in a container, you add some water with nutrient, all right? You put it under the light, good pH and everything, and it will start to grow. And whether this lag phase is slow or fast, it all depends on a couple of things. Okay. If you, let's say, add in a microalgae which is already dying, already in bad condition, then it will grow very slowly here. It will take time to go up. The lag phase is longer. If you add here microalgae, which is essentially new cells or young cells from the exponential stage at another container, it will tend to have very short lag phase. It may go up very fast. So from the lag phase, it goes up, the exponential phase, and then you have a lag, or what do you call this, the declining growth, stationary, and after a while when you have lack of nutrient or light, okay, or maybe some contamination or toxicity, then it will die, and that is the death phase, and that is called the stage number 5. All right, so I see someone joining the class. There you go, hold on. All right. So when you want to grow your microalgae in containers, okay, when you upscale from small density, from small scale to a bigger scale, from small container to bigger container, let's say you start with 50 milliliter, what we call as inoculum, and then go up to 500, go up to 1000, and so on and so forth. You need to make sure you transfer the microalgae at the phase in what we call as exponential phase. Exponential phase, which is this, and there are some ways to actually maintain your microalgae culture. You can do the daily harvesting to replenish the nutrient, all right, and always maintain it at exponential phase. And then what do you call this? Once you reach the declining growth phase, stationary phase, and so on, due to nutrient very low nutrient, due to possible toxic properties, okay, metabolites and poor digestibility, then you start declining growth. All right. Now this you will see, you will observe in your growth, your microalgae culture when we do it in the lab later on. So in the lab, when we do a face-to-face meeting, and then you'll find out, you can find out how it looks like in terms of the growth of your microalgae. All right. we'll do this later on, this particular growth phase of microalgae. We'll try to do this in the lab and see how it looks like, okay? Now, for purpose of production, microalgae, normally you can collect it from the wild, or you can buy it from specialized collection, or you buy it from other universities. You know, some universities sell microalgae, all right? So let's say you want to start your own hatchery, and you want to produce your diatoms such as Skeletonema or Chitoceras. You want to start that, right? You have your own hatchery, or you want to start hatchery for your fish, marine fish, and you want nanochloropsis. So before starting your fish hatchery, your shrimp hatchery, you want to buy or you want to get supply of your microalgae, all right? So where do you get them from? Well, you can get them from the wild, collect them from the sea, from the freshwater lakes and rivers and so on. Of course, you can do that. Or you can buy from other hatcheries. Some hatcheries do sell their microalgae. So you call your friend who is also a hatchery operator. Hey, you know, I want to buy some microalgae. Do you sell those? Do you sell some of those? Do you sell some Skeletonema or Chitoceras? But some of your good friends may not sell to you. They just say, okay, you take some, take some. I'll give you for free your microalgae inoculum or starter microalgae population. Okay, so starting the starter population or the inoculum, they call it, you can get sometimes for free from others in the industry. Or if you want some really very good culture, or maybe some without contamination, or maybe some unique microalgae, you might want to get it from universities who produce them. I know of some university in Malaysia who produce microalgae regularly, and you can even buy them from outside the country, okay? You can buy them and import them to Malaysia as well. No issue with that, okay? For research purposes, okay. For purpose of running your hatchery, it's okay also, all right? Let me see. Okay, there's someone joining the class. Okay, Lorita, welcome to the class, Lorita. Okay. So when you want to grow microalgae, you just need to heat the sources of contamination, which is your culture medium, your air, culture vessels, okay, your inoculum itself, the initial population of microalgae that you use to upscale. These are all happening all the time, all right? The stock contamination. So you need to be aware of this, especially when you start growing them in the lab. And these are ways and means to reduce your contamination, sterilization, UV radiation, autoclaving, what have you. Quite a number of ways people are doing this, all right? So in the lab, later on, we will go through this a bit more, and we will teach you how to reduce contamination in the lab, and also how to reduce your contamination in practical sense as you do upscaling and you run a lab or when you run a hatchery, okay? So reducing the contamination of microalgae in the lab is quite different when you want to use it outside in a bigger scale, all right? For commercial purposes, it's a bit different in a way of reducing the contamination, okay? Depends on what is the density and also the abundance of your in containers, small or large, and then you will uh, reduce the contamination using some approaches which is more relevant to it. Okay, so this is a paropsis. For naturally like us, which is plankton, which become live food for your zooplankton or for your rotifer. So the rotifer now becomes food for the fish larvae. So first you fatten your rotifer, give it a lot of nutrient from the microalgae that you culture, then you feed it to your fish. All right. So in this case, when you want to grow your grouper fish, okay, or your ikan siakap or your sea bass, you need to culture two types of live food. One is the microalgae. In this case, is the nanochloropsis. The other one is your rotifer, which is a zooplankton. All right. So culture both, so that you can make sure that your fish is well fed and grow fast, especially in the larval stage. This is an example of how you upscale your microalgae production. This is called batch culture. You start with a test tube, you increase it 2 liter flask, okay? After a few days, you increase further to secondary flask, carboys, a bigger container, all right? And then to fiberglass tank. Sometimes you can use, it doesn't matter, any kind of tanks will do, fiberglass, canvas tank, or maybe cement tank. You can do all those, no issue at all. All right. So essentially, when you do batch culture, upscaling, you start from a small quantity like this, and then you go to a bigger quantity all the way down to the last volume, which is your mass culture, before you give it to your fish, or you give it, let's say in this case, if you want to grow a group of fish, you grow up to this scale, give it to your rotifer. All right. So from here to here takes a lot of time, right? So it can be like here 10 days, 3 to 6 days. Sorry, from here 10 to 14 days, almost 2 weeks. This is like a week, another week, and so on and so forth. So this means that if you want to grow microalgae to feed your fish and your shrimp, you need time. You need to start early. You cannot do it immediately. Let's say your fish is already matured, it has undergone spawning and produced the eggs and hatch. And then after hatching, the larvae grows well with the yolk sac, absorb the yolk sac. Let's say after 1 week, the fish larvae have absorbed all the yolk sac. All right. Once the fish larvae have absorbed all the yolk sac, then it needs to eat, right? And you cannot just do the feeding there and then, or prepare your microalgae there and then, or prepare your rotifer just like that. You need to get everything ready a few weeks before you feed the fish larvae. So the timing is very important, okay? So this is also one example of upscaling from stock culture to 150 ml, or sometimes you to get the stock culture, you start with your inoculum. Remember the term inoculum, this one, right? Inoculum, you can get it from your own, from other places, so you can buy it, it's okay. So from the inoculum, okay, you put it into 250 ml beaker in this case, all right, or flask rather. This is flask, and it become your stock culture. As it grow and multiply, you go to a bigger culture, all right, starter culture here, and then after a few more days, sometimes even up to 1 to 2 weeks, you have intermediate culture, which is already 4 to 20 liter, and then after that, you add to your large scale culture more than 50 liter, can be about 1,000 liters, 2,000, depends lah on how you want to do it. So all this takes time, okay? All this takes time and must be done prior to your fish spawning. Get everything done. Once your fish starts spawning, you want to do this, it becomes too late already. Okay, so that is why it's very important in the hatchery for the hatchery technician or the manager to understand how the timing of when to feed your fish larvae and prepare accordingly much earlier. If not, you cannot get your production going. Okay. So usually when you use batch culture, you start with inoculum. It corresponds to a scaling of about 2 to 10% of the final volume. For example, 2 to 10%, let's say in this case, you start with 250 ml, right? 250 ml, you want to give it to start 250, add on to 4 liter. Now this is about what? Okay, 10% of 4 liter is 400 ml. So this is about 6%. Okay, 6%, you add 6% of 4 liter. Okay, as I said just now, it's anywhere between 2 to 10%, right? So 6% is about right. Okay, and then 4 liter to 20 liter. Okay, that is about 20. That's about 20%, right? Okay, 4 to 20, 4 liter to 20 liter, 20 liter to more than 50 liter. All right. Okay, so this is an example of the flask that you use to grow your microalgae. This is a 200 ml flask. Okay, and in this hatchery, this is a hatchery in Terengganu. This is a shrimp hatchery. And the brownish color here is microalgae of the group or class Bacillariophyceae, which is your Chaetoceros. Here, Chaetoceros, right? This flask is still very new. This is a bit older and it's growing well. You can see the brownish color. This is the light source. In this lab, okay, they grow Chaetoceros to feed the the shrimp larva. This is in a shrimp hatchery. Hatchery udang ni. Dia menghasilkan skeletonema sebagai makanan hidup. Okay. Untuk udang. Alright, so from here, 200 ml, it goes up to 2 liter flask. You see all this? Look at the color. This is the color of Chaetoceros, which is of a good abundance. The density is good enough. This is a good density of Chaetoceros. Nampak yang bawah ni? Warna dia coklat gelap sikit. Okay, dah cukup padat lah ni bawah ni. Yang ini baru lagi. Up here still new. Color still changing from almost green to brown. This one is already mature enough. You can transfer this to a bigger tank. Right? So why brown in color? Because diatoms tend to become brownish or golden brown in color. All right. Now this, your Nanocoropsis is green in color. This is a different class of microalgae. This is Prasinophyceae. This is for your Nanocoropsis. So that's why it is green. And this is your Chaetoceros, which is a diatom, and it becomes a brownish or golden brown color. All right, so from 200 ml bottle to 2 liters, and then from 2 liters to 500 liter tanks. Okay, sometimes you may have to put in a few bottles here, not necessarily just one. Get a few bottles here to transfer into this 500 liter tank. Put in new water with nutrient and bring it up with some lighting. You can get more of the diatom here. All right, so all this tank has now the upscaled Chaetoceros to be fed to your shrimp larva. Okay. Now, if you notice also, the tank here is a bit translucent. Okay. You can see that the light can go through, right? On the left and the right side, everywhere around here. This is to make sure that the microalgae has enough light within the tank and also from the top. This is a bigger tank, which is about 20,000 liter or 20-ton tank, where we do mass production of microalgae. Look at this. Chitosaurus species, brown in color, as mentioned to you just now. Okay. And you need this bubbling, right? Why is this bubbling important? Well, there is aeration and mixing. Okay, I mentioned to you earlier the importance of having some mixing and also an aeration. Okay, so that all the microalgae have access to nutrient, have access to light. All right. So very important to get some air bubble blowing down here as well. Look at this. This is another kind of diatom called Thalassiosira, also in a shrimp hatchery. Okay, you still use bubbles here to inoculate, to what do you call this? To maintain the culture. All right. This is your Skeletonema, or I think this is Chitosaurus. Sorry. This is Chitosaurus in a shrimp lab. This lab is in the Perak. Okay, and there's a place called Teluk Senangin. Teluk Senangin is in Perak near Lumut there. Okay, after Setiawan, after Setiawan there near Lumut, you have this particular company producing microalgae in the lab to be fed to the shrimp larva. All right. This is mass culture of microalgae, mass culture of diatoms. In this case, this company produces Chitosaurus. Okay, when you see the water like very dark brown like this, it means that your Chitosaurus is very abundant. All right, banyak Chitosaurus dia ni. Okay, see all these tanks, all filled up with Chitosaurus diatom. All right. And sometimes you can use cylinders like this, and you know microalgae comes in different varieties, different colors, and the color will tell you what species of microalgae there is here. This is the culture of Nanochloropsis. This is a backyard culture. This was taken in Terengganu. I took this photo behind a guy's hatchery where he produces grouper fry, grouper and siakap. And this is Nanochloropsis that they grow. And this Nanochloropsis will be later on transferred to a tank and to be fed to the rotifer. Remember, small fish, marine fish normally will eat or will consume zooplankton. But zooplankton will need to be fed first of all, or fatten it by your microalgae, right? So this is the Nanochloropsis that they use back in the backyard to feed to the rotifer. This is an example of outdoor pond, outdoor tanks where they grow Nanochloropsis also in the fish hatchery. Look at this color; it's green, right? Okay, it's different from this. Well, this is brown. Look at that, very brown. Huh? So this is diatom. This is more for marine fish, marine shrimp, udang laut. Diatom. Okay, Chitosaurus. This is for marine fish. In this case, it is your grouper. Okay. Now you grow this to feed the diatom. They grow this to feed the rotifer. And rotifer will then be fed to the fish. This is another outdoor tank. You look at this color, brownish, right? So this is your Chitosaurus. This is your Nanochloropsis. Different color, different species. This is Nanochloropsis. Again, in a mass culture, big quantity culture, and you use here in this case you use just a normal agriculture grade fertilizer. All right, to use all this. This tank is about I think about 5-ton tank, meaning 5,000 liters of water. Okay. These are some examples of outdoor pond or tanks where they grow microalgae in the what we call as photobioreactors or in very large quantity, in very high density. Okay, this is production of spirulina. Use it using this sort of method. Okay, it has to be a very good place, dry, no rain, and so on. Then you get consistent production. Okay. Some of the in fact, all of the pharmaceutical products that we buy, like the chlorella tablet, chlorella capsule, spirulina capsule, spirulina tablet, all those are grown in this sort of condition, this sort of environment. Okay, they don't get this from the wild. They actually culture spirulina. Okay, and they culture chlorella. After that, they will process the microalgae to make it into pills, all right, to make it into capsules for human consumption. Okay. Okay, I want to stop there. And then we'll continue with this later on. Stop at the picture about this one, and then if you have any questions, feel free to ask me right now. Okay. Ada persoalan tak? Is it okay? Boleh. You learn something today on microalgae culture. All right. I hope I've given you enough information to give you some idea on how you can grow microalgae. Essentially, this is only like information, from notes and so on, but not really a hands-on thing yet. Okay. So we'll come back after the first week of November, that we can do all the necessary hands-on practical work. All right. That will be more interesting for all of you. You can see it. You can see your microalgae growing in your flask. First, the culture will be very transparent. You don't see anything, just very little yellow color or brown. And then slowly, as the days go by, you can see it brownish, greenish, It's going to be a bit more exciting. And then you can even feed them to your rotifer as we move along. That will be better, Hopefully, there's no longer any issue of coming back to class or to face-to-face meeting. Okay. All right. So I'd like to welcome some new students who joined the class just now. Okay, I will get to know you a bit more later on Friday. So do join us on Friday if you just joined the class today. Friday kelas kita pukul berapa? What time? I always forget about this. 3 o'clock, is it? Friday?
Yes.
Yes, lah.
Okay, 3 o'clock. Yeah, so you're going to have quite a number of students in this class. As I mentioned to the first group, earlier group just now, we have altogether about 43 students. All right, so you have to, I will add you in. Okay, let me see. Do we have a WhatsApp group tau ni? We have a WhatsApp group. And then if you have a phone number, whatever, you can, or if you guys know how to get in touch with anyone in this group, okay, we can add you into the WhatsApp for this particular class. Yeah. Kalau you boleh ada contact siapa-siapa dalam group ni, okay, you kenal any of the seniors here, then you can join lah and the WhatsApp group. If not, I try to also put you in from our connection, from our communication earlier. All right, now your notes, okay, I try to get it up to Putra Blast. Okay, as I said earlier, if there are issues with Putra Blast, I will just send it to your WhatsApp. As long as you can get the notes, I mean, it's okay lah kan. All right. either Putra Blast ke, or either WhatsApp ke. For the last few groups I had last semester,